Section 13.1 Prokaryotic Diversity. By the end of this section, you will be able to describe the evolutionary history of prokaryotes, describe the basic structure of a typical prokaryote, identify bacterial diseases that cause historically important plagues and epidemics, and describe the use of prokaryotes in food processing and bioremediation. Prokaryotes are present everywhere. They cover every imaginable surface where there is sufficient moisture, and they live on and inside of other living things. There are more prokaryotes inside and on the exterior of the human body than there are human cells in the body. Some prokaryotes thrive in environments that are inhospitable for most other living things. Prokaryotes recycle nutrients, essential substances such as carbon and nitrogen, and they drive the evolution of new ecosystems, some of which are natural while others are man-made. Prokaryotes have been on Earth since long before multicellular life appeared. Prokaryotic diversity. The advent of DNA sequencing provided immense insight into the relationships and origins of prokaryotes that were not possible using traditional methods of classification. A major insight identified two groups of prokaryotes that were found to be as different from each other as they were from eukaryotes. This recognition of prokaryotic diversity forced a new understanding of the classification of all life and brought us closer to understanding the fundamental relationships of all living things, including ourselves. Early life on Earth. When and where did life begin? What were the conditions on Earth when life began? Prokaryotes were the first forms of life on Earth, and they existed for billions of years before plants and animals appeared. Earth is about 4.54 billion years old. This estimate is based on evidence from the dating of meteorite material, since surface rocks on Earth are not as old as Earth itself. Most rocks available on Earth have undergone geological changes that make them younger than Earth itself. Some meteorites are made of the original material in the solar disk that formed the objects of the solar system, and they have not been altered by the processes that altered rocks on Earth. Thus, the age of meteorites is a good indicator of the age of the formation of Earth. The original estimate of 4.54 billion years was obtained by Claire Patterson in 1956. His meticulous work has since been corroborated by ages determined from other sources, all of which point to an Earth age of about 4.54 billion years. Early Earth had a very different atmosphere than it does today. Evidence indicates that during the first 2 billion years of Earth's existence, the atmosphere was anoxic, meaning there was no oxygen. Therefore, only those organisms that can grow without oxygen, anaerobic organisms, were able to live. Organisms that convert solar energy into chemical energy are called phototrophs. Phototrophic organisms that required an organic source of carbon appeared within 1 billion years of the formation of Earth. Then, cyanobacteria, also known as blue-green algae, evolved from these simple phototrophs 1 billion years later. Cyanobacteria are able to use carbon dioxide as a source of carbon. Cyanobacteria began the oxygenation of the atmosphere. The increase in oxygen concentration allowed the evolution of other life forms. Figure 13.2. This hot spring in Yellowstone National Park flows toward the foreground. Cyanobacteria in the spring are green, and as the water flows down the heat gradient, the intensity of the color increases because the cell density increases. The water is cooler at the edges of the stream than in the center, causing the edges to appear greener. Before the atmosphere became oxygenated, the planet was subjected to strong radiation. Thus, the first organisms would have flourished where they were more protected, such as in ocean depths or beneath the surface of Earth. At this time, too, strong volcanic activity was common on Earth, so it is likely that these first organisms, the first prokaryotes, were adapted to very high temperatures. These are not the typical temperate environments in which most life flourishes today. Thus, we can conclude that the first organisms that appeared on Earth were likely able to withstand harsh conditions. Microbial mats may represent the earliest forms of life on Earth, and there is fossil evidence of their presence starting about 3.5 billion years ago. A microbial mat is a large biofilm, a multi-layered sheet of prokaryotes, including most bacteria, or including mostly bacteria, but also archaea. Microbial mats are a few centimeters thick, and they typically grow on moist surfaces. Their various types of prokaryotes carry out different metabolic pathways, and for this reason, they reflect various colors. Prokaryotes in a microbial mat are held together by a gummy-like substance that they secrete. The first microbial mats likely obtained their energy from hydrothermal vents. A hydrothermal vent is a fissure in Earth's surface that releases geothermally heated water. With the evolution of photosynthesis about 3 billion years ago, some prokaryotes in microbial mats came to use a more widely available energy source, sunlight, whereas others were still dependent on chemicals from hydrothermal vents for food. Figure 13.3a. This microbial mat grows over a hydrothermal vent in the Pacific Ocean. Chimneys, such as the one indicated by the arrow, allow gases to escape. B, the image on the right, this photo shows stromatolites that are nearly 1.5 billion years old, found in Glacier National Park, Montana. Fossilized microbial mats represent the earliest record of life on Earth. A stromatolite is a sedimentary structure formed when minerals are precipitated from water by prokaryotes in a microbial mat. Stromatolites form layered rocks made of carbonate or silica. Although most stromatolites are artifacts from the past, there are places on Earth where stromatolites are still forming. For example, living stromatolites have been found in the Anza Borrego Desert State Park in San Diego County, California. Efforts to understand the earliest microbial mats may also have implications on the search for life elsewhere. Nora Nofke, who identified many of the features used in dating and categorizing microbially produced sedimentary structures, emphasizes that similar formations could be used to find evidence of life on Mars. When analyzing photos taken by the NASA Curiosity rover, Nofke showcased the similarities between fossilized microbial mats found on the two planets. Some prokaryotes are able to thrive and grow under conditions that would kill a plant or animal. Bacteria and archaea that grow under extreme conditions are called extremophiles, meaning lover of extremes. Extremophiles have been found in extreme environments of all kinds, including the depths of the oceans, hot springs, the Arctic and the Antarctic, very dry places, deep inside Earth, harsh chemical environments, and high radiation environments. Extremophiles give us a better understanding of prokaryotic diversity and open up the possibility of the discovery of new therapeutic drugs or industrial applications. They have also opened up the possibility of finding life in other places in the solar system, which have harsher environments than those typically found on Earth. Many of these extremophiles cannot survive in moderate environments. Biofilms. Until a couple of decades ago, microbiologists thought of prokaryotes as isolated entities living apart. This model, however, does not reflect the true ecology of prokaryotes, most of which prefer to live in communities where they can interact. A biofilm is a microbial community held together in a gummy-textured matrix consisting primarily of polysaccharides secreted by the organisms, together with some proteins and nucleic acids. Biofilms grow attached to surfaces. Some of the best studied biofilms are composed of prokaryotes, although fungal biofilms have also been described. Biofilms are present almost everywhere. They cause the clogging of pipes and readily colonize surfaces in industrial settings. They have played roles in recent large-scale outbreaks of bacterial contamination of food. Biofilms also colonize household surfaces, such as kitchen counters, cutting boards, sinks, and toilets. Interactions among the organisms that populate a biofilm, together with their protective environment, make these communities more robust than our free-living or planktonic prokaryotes. Overall, biofilms are very difficult to destroy because they are resistant to many of the common forms of sterilization. Characteristics of prokaryotes. There are many differences between prokaryotic and eukaryotic cells. However, all cells have four common structures: a plasma membrane that functions as a barrier for the cell and separates the cell from its environment, cytoplasm, a jelly-like substance inside the cell, genetic material, DNA and RNA, and ribosomes, where protein synthesis takes place. Prokaryotes come in various shapes, but many fall into three categories: cocci, or spherical, bacilli, or rod-shaped, and spirilla, spiral-shaped. Figure 13.4. Many prokaryotes fall into three basic categories based on their shape. On the left, A, cocci or spherical. In the middle, B, bacilli or rod-shaped. And on the right, C, spirilla or spiral-shaped. The prokaryotic cell. Recall that prokaryotes are unicellular organisms that lack organelles surrounded by membranes. Therefore, they do not have a nucleus, but instead have a single chromosome, a piece of circular DNA located in an area of the cell called the nucleoid. Most prokaryotes have a cell wall laying outside the plasma membrane. The composition of the cell wall differs significantly between the domains bacteria and archaea, and their cell walls also differ from the eukaryotic cell walls found in plants and fungi. The cell wall functions as a protective layer and is responsible for the organism's shape. Some other structures are present in some prokaryotic species, but not in others. For example, the capsule, found in some species, enables the organism to attach to surfaces and protects it from dehydration. Some species may also have flagella, or singular flagellum, used for locomotion, and pili, singular pilus, used for attachment to surfaces and to other bacteria for conjugation. Plasmids, which consist of small circular pieces of DNA outside of the main chromosome, are also present in many species of bacteria. Figure 13.5 shows the features of a typical bacterium cell wall. Both bacteria and archaea are types of prokaryotic cells. They differ in the lipid composition of their cell membranes and in the characteristics of their cell walls. Both types of prokaryotes have the same basic structures, but these are built from different chemical components that are evidence of an ancient separation of their lineages. The archaeal plasma membrane is chemically different from the bacterial membrane. Some archaeal membranes are lipid monolayers instead of phospholipid bilayers. The cell wall. The cell wall is a protective layer that surrounds some prokaryotic cells and gives them shape and rigidity. It is located outside the cell membrane and prevents osmotic lysis, bursting caused by increasing volume. The chemical compositions of the cell walls vary between archaea and bacteria, as well as between bacterial species. Bacterial cell walls contain peptidoglycan, composed of polysaccharide chains cross-linked to peptides. Bacteria are divided into two major groups, gram-positive and gram-negative, based on their reaction to a procedure called gram staining. The different bacterial responses to the staining procedure are caused by cell wall structure. Gram-positive organisms have a thick wall consisting of many layers of peptidoglycan. Gram-negative bacteria have a thinner cell wall composed of a few layers of peptidoglycan and additional structures surrounded by an outer membrane. Figure 13.6. Bacteria are divided into two major groups, gram-positive and gram-negative. Both groups have a cell wall composed of peptidoglycans. In gram-positive bacteria, the wall is thick, whereas in gram-negative bacteria, the wall is thin. In gram-negative bacteria, the cell wall is surrounded by an outer membrane. Archaeal cell walls do not contain peptidoglycan. There are 4 different types of archaeal cell walls. One type is composed of pseudopeptidoglycan. The other 3 types of cell walls contain polysaccharides, glycoproteins, and surface layer proteins known as S layers. Reproduction. Reproduction in prokaryotes is primarily asexual and takes place by binary fission. Recall that the DNA of a prokaryote exists usually as a single circular chromosome. Prokaryotes do not undergo mitosis. Rather, the chromosome loop is replicated, and the 2 resulting copies attached to the plasma membrane move apart as the cell grows in a process called binary fission. The prokaryote, now enlarged, is pinched inward at its equator, and the 2 resulting cells, which are clones, separate. Binary fission does not provide an opportunity for genetic recombination, but prokaryotes can alter their genetic makeup in 3 ways. Binary fission, as a way of reproduction, does not provide an opportunity for genetic recombination and increased genetic variability. However, prokaryotes can alter their genetic makeup by 3 mechanisms of obtaining exogenous DNA. In a process called transformation, the cell takes in DNA found in its environment that is shed by other prokaryotes, alive or dead. A pathogen is an organism that causes a disease. If a non-pathogenic bacterium takes up DNA from a pathogen and incorporates the new DNA in its own chromosome, it too may become pathogenic. In transduction, bacteriophages, the viruses that infect bacteria, move DNA from one bacterium to another. Archaea have a different set of viruses that infect them and translocate genetic material from one individual to another. During conjugation, DNA is transferred from one prokaryote to another by means of a pilus that brings the organisms into close contact with one another. The DNA transferred is usually a plasmid, but parts of the chromosome can also be moved. Cycles of binary fission can be very rapid, on the order of minutes for some species. This short generation time, coupled with mechanisms of genetic recombination, result in the rapid evolution of prokaryotes, allowing them to respond to environmental changes, such as the introduction of an antibiotic, very quickly. How prokaryotes obtain energy and carbon. Prokaryotes are metabolically diverse organisms. Prokaryotes fill many niches on Earth, including being involved in nutrient cycles such as the nitrogen and carbon cycles, decomposing dead organisms, and growing and multiplying inside living organisms, including humans. Different prokaryotes can use different sources of energy to assemble macromolecules from smaller molecules. Phototrophs obtain their energy from sunlight. Chemotrophs obtain their energy from chemical compounds. Bacterial diseases in humans. Devastating pathogen-borne diseases and plagues, both viral and bacterial in nature, have affected and continue to affect humans. It is worth noting that all pathogenic prokaryotes are bacteria. There are no known pathogenic archaea in humans or any other organisms. Pathogenic organisms evolved alongside humans. In the past, the true cause of these diseases was not understood, and some cultures thought that diseases were a spiritual punishment or were mistaken about material causes. Over time, people came to realize that staying apart from afflicted persons, improving sanitation, and properly disposing of the corpses and personal belongings of victims of illness reduced their own chances of getting sick. There are records of infectious diseases as far back as 3000 BC. A number of significant pandemics caused by bacteria have been documented over several hundred years. Some of the largest pandemics led to the decline of cities and cultures. Many were zoonoses that appeared with the domestication of animals, as in the case of tuberculosis. A zoonosis is a disease that infects animals but can be transmitted from animals to humans. Infectious diseases remain among the leading causes of death worldwide. Their impact is less significant in many developed countries, but they are important determiners of mortality in developing countries. The development of antibiotics did much to lessen the mortality rates from bacterial infections, but access to antibiotics is not universal, and the overuse of antibiotics has led to the development of resistant strains of bacteria. Public sanitation efforts that dispose of sewage and provide clean drinking water have done as much or more than medical advances to prevent deaths caused by bacterial infections. In 430 BC, the plague of Athens killed 1/4 of the Athenian troops that were fighting the Great Peloponnesian War. The disease killed 1/4 of the population of Athens in over 4 years and weakened Athens' dominance and power. The source of the plague may have been identified recently when researchers from the University of Athens were able to analyze DNA from teeth recovered from a mass grave. The scientists identified nucleotide sequences from a pathogenic bacterium that causes typhoid fever. From 541 to 750 AD, an outbreak called the Plague of Justinian, likely a bubonic plague, eliminated, by some estimates, 1/4 to 1/2 of the human population. The population in Europe declined by 50% during this outbreak. Bubonic plague would decimate Europe more than once. One of the most devastating pandemics was the Black Death, 1346 to 1361, which is believed to have been another outbreak of bubonic plague caused by the bacterium Yersinia pestis. This bacterium is carried by fleas living on black rats. The Black Death reduced the world's population from an estimated 450 million to about 350 to 375 million. Bubonic plague struck London hard again in the mid-1600s. There are still approximately 1,000 to 3,000 cases of plague globally each year. Although contracting bubonic plague before antibiotics meant almost certain death, the bacterium responds to several types of modern antibiotics, and mortality rates from plague are now very low. Over the centuries, Europeans developed resistance to many infectious diseases. However, European conquerors brought disease-causing bacteria and viruses with them when they reached the Western Hemisphere, triggering epidemics that completely devastated populations of Native Americans who had no natural resistance to many European diseases. The antibiotic crisis. The word antibiotic comes from the Greek anti, meaning against, and bios, meaning life. An antibiotic is an organism-produced chemical that is hostile to the growth of other organisms. Today's news and media often address concerns about an antibiotic crisis. Are antibiotics that were used to treat bacterial infections easily treatable in the past becoming obsolete? Are there new superbugs, bacteria that have evolved to become more resistant to our arsenal of antibiotics? Is this the beginning of the end of antibiotics? All of these questions challenge the healthcare community. One of the main reasons for resistant bacteria is the overuse and incorrect use of antibiotics, such as not completing a full course of prescribed antibiotics. The incorrect use of an antibiotic results in the natural selection of resistant forms of bacteria. The antibiotic kills most of the infecting bacteria, and therefore only the resistant forms remain. These resistant forms reproduce, resulting in an increase in the proportion of resistant forms over non-resistant ones. Another problem is the excessive use of antibiotics in livestock. The routine use of antibiotics in animal feed promotes bacterial resistance as well. In the United States, 70% of the antibiotics produced are fed to animals. The antibiotics are not used to prevent disease, but to enhance production of their products. Staphylococcus aureus, often called staph, is a common bacterium that can live in and on the human body, which usually is easily treatable with antibiotics. A very dangerous strain, however, has made the news over the past few years. This strain, methicillin-resistant Staphylococcus aureus, or MRSA, is resistant to many commonly used antibiotics, including methicillin, amoxicillin, penicillin, and oxacillin. While MRSA infections have been common among people in healthcare facilities, it is appearing more commonly in healthy people who live or work in dense groups, like military personnel and prisoners. The Journal of the American Medical Association reported that among MRSA-afflicted persons in healthcare facilities, the average age is 68 years, while people with community-associated MRSA, CA-MRSA, has an average age of 23 years. Figure 13.7. This scanning electron micrograph shows methicillin-resistant Staphylococcus aureus bacteria, commonly known as MRSA. In summary, society is facing an antibiotic crisis. Some scientists believe that after years of being protected from bacterial infections by antibiotics, we may be returning to a time in which a simple bacterial infection could again devastate the human population. Researchers are working on developing new antibiotics, but few are in the drug development pipeline, and it takes many years to generate an effective and approved drug. Foodborne diseases. Prokaryotes are everywhere. They readily colonize the surface of any type of material, and food is not an exception. Outbreaks of bacterial infection related to food consumption are common. A foodborne disease, colloquially called food poisoning, is an illness resulting from the consumption of food contaminated with pathogenic bacteria, viruses, or other parasites. Although the United States has one of the safest food supplies in the world, the Center for Disease Control and Prevention, the CDC, has reported that 76 million people get sick, more than 300,000 are hospitalized, and 5,000 Americans die each year from foodborne illness. The characteristics of foodborne illness have changed over time. In the past, it was relatively common to hear about sporadic cases of botulism, the potentially fatal disease caused by a toxin from the anaerobic bacterium Clostridium botulinum. A can, jar, or package created a suitable anaerobic environment where Clostridium could grow. Proper sterilization and canning procedures have reduced the incidence of this disease. Most cases of foodborne illnesses are now linked to produce contaminated by animal waste. For example, there have been serious produce-related outbreaks associated with raw spinach in the United States and with vegetable sprouts in Germany. The raw spinach outbreak in 2006 was produced by the bacterium E. coli strain O157H7. Most E. coli strains are not particularly dangerous to humans. Indeed, they live in our large intestine, but O157H7 is potentially fatal. Figure 13.8. On the left, locally grown vegetable sprouts were the cause of a European E. coli outbreak that killed 31 people and sickened about 3,000 in 2010. On the right, E. coli are shown here in scanning electron micrograph. The strain of E. coli that caused a deadly outbreak in Germany is a new one, not involved in any previous E. coli outbreaks. It has acquired several antibiotic resistance genes and specific genetic sequences involved in aggregation ability and virulence. It has recently been sequenced. All types of food can potentially be contaminated with harmful bacteria of different species. Recent outbreaks of salmonella, reported by the CDC, occurred in foods as diverse as peanut butter, alfalfa sprouts, and eggs. Career connection. Epidemiologist. Epidemiology is the study of the occurrence, distribution, and determinants of health and disease in a population. It is, therefore, related to public health. An epidemiologist studies the frequency and distribution of diseases within human populations and environments. Epidemiologists collect data about a particular disease and track its spread to identify the original mode of transmission. They sometimes work in close collaboration with historians to try to understand the way a disease evolved geographically and over time, tracking the natural history of pathogens. They gather information from clinical records, patient interviews, and any other available means. That information is used to develop strategies and design public health policies to reduce the incidence of a disease or to prevent its spread. Epidemiologists also conduct rapid investigations in case of an outbreak to recommend immediate measures to control it. Epidemiologists typically have a graduate level education. An epidemiologist often has a bachelor's degree in some field and a master's degree in public health. Many epidemiologists are also physicians and have an MD, or they have a PhD in an associated field, such as biology or epidemiology. Beneficial prokaryotes. Not all prokaryotes are pathogenic. On the contrary, pathogens represent only a very small percentage of the diversity of the microbial world. In fact, our life and all life on this planet would not be possible without prokaryotes. Prokaryotes and food and beverages. According to the United Nations Convention on Biological Diversity, biotechnology is any technological application that uses biological systems, living organisms, or derivatives thereof to make or modify products or processes for specific use. The concept of specific use involves some sort of commercial application. Genetic engineering, artificial selection, antibiotic production, and cell culture are current topics of study in biotechnology. However, humans have used prokaryotes to create products before the term biotechnology was even coined, and some of the goods and services are as simple as cheese, yogurt, sour cream, vinegar, cured sausage, sauerkraut, and fermented seafood that contains both bacteria and archaea. Cheese production began around 4,000 years ago when humans started to breed animals and process their milk. Evidence suggests that cultured milk products like yogurt have existed for at least 4,000 years. Using prokaryotes to clean up our planet. Bioremediation. Microbial bioremediation is the use of prokaryotes or microbial metabolism to remove pollutants. Bioremediation has been used to remove agricultural chemicals, pesticides, and fertilizers that leach from soil into groundwater. Certain toxic metals, such as selenium and arsenic compounds, can also be removed from water by bioremediation. The reduction of SeO4 2- to SeO3 2- and to SeO, metallic selenium, is a method used to remove selenium ions from water. Mercury is an example of a toxic metal that can be removed from an environment by bioremediation. Mercury is an active ingredient of some pesticides. It is used in industry and is also a byproduct of certain industries, such as battery production. Mercury is usually present in very low concentrations in natural environments, but it is highly toxic because it accumulates in living tissues. Several species of bacteria can carry out the biotransformation of toxic mercury into non-toxic forms. These bacteria, such as Pseudomonas aeruginosa, can convert Hg2+ to Hg0, which is non-toxic to humans. Probably one of the most useful and interesting examples of the use of prokaryotes for bioremediation purposes is the cleanup of oil spills. The importance of prokaryotes to petroleum bioremediation has been demonstrated in several oil spills in recent years, such as the Exxon Valdez spill in Alaska in 1989, the Prestige oil spill in Spain in 2002, and the spill into the Mediterranean from a Lebanon power plant in 2006. And more recently, the BP oil spill in the Gulf of Mexico in 2010. To clean up these spills, bioremediation is promoted by adding inorganic nutrients that help bacteria already present in the environment to grow. Hydrocarbon-degrading bacteria feed on the hydrocarbons in the oil droplet, breaking them into inorganic compounds. Some species, such as Alkinovorax borchimensis, produce surfactants that solubilize the oil, while other bacteria degrade the oil into carbon dioxide. In the case of oil spills in the ocean, ongoing natural bioremediation tends to occur inasmuch as there are oil-consuming bacteria in the ocean prior to the spill. Under ideal conditions, it has been reported that up to 80% of the non-volatile components in oil can be degraded within 1 year of the spill. Other oil fractions containing aromatic and highly branched hydrocarbon chains are more difficult to remove and remain in the environment for longer periods of time. Researchers have genetically engineered other bacteria to consume petroleum products. Indeed, the first patent application for a bioremediation application in the United States was for a genetically modified oil-eating bacterium. Figure 13.10. On the left, cleaning up after the Valdez spill in Alaska, the workers hosed oil from beaches and then used a floating boom to corral the oil, which was finally skimmed from the water surface. Some species of bacteria are able to solubilize and degrade the oil. On the right, one of the most catastrophic consequences of oil spills is the damage to fauna. Prokaryotes in and on the body. Humans are no exception when it comes to forming symbiotic relationships with prokaryotes. We are accustomed to thinking of ourselves as single organisms, but in reality, we are walking ecosystems. There are 10 to 100 times as many bacterial and archaeal cells inhabiting our bodies as we have cells in our bodies. Some of these are in mutually beneficial relationships with us, in which both the human host and the bacterium benefit, while some of the relationships are classified as commensalism, a type of relationship in which the bacterium benefits and the human host is neither benefited nor harmed. Human gut flora lives in the large intestine and consists of hundreds of species of bacteria and archaea, with different individuals containing different species mixes. The term flora, which is usually associated with plants, is traditionally used in this context because bacteria were once classified as plants. The primary functions of these prokaryotes for humans appears to be metabolism of food molecules that we cannot break down, assistance with the absorption of ions by the colon, synthesis of vitamin K, training of the infant immune system, maintenance of the adult immune system, maintenance of the epithelium of the large intestine, and formation of a protective barrier against pathogens. The surface of the skin is also coated with prokaryotes. The different surfaces of the skin, such as the underarms, the head, and the hands, provide different habitats for different communities of prokaryotes. Unlike with gut flora, the possible beneficial roles of skin flora have not been well studied. However, the few studies conducted so far have identified bacteria that produce antimicrobial compounds as probably responsible for preventing infections by pathogenic bacteria. As they would in any ecosystem, the organisms in the microbiome are subject to selection and evolution. Simply to survive through changes in the human body requires a degree of adaptability and resistance. Because humans and other animals, such as livestock, ingest more and more antibiotics, the bacteria within our bodies are becoming resistant to these medicines. Abigail A. Salyers, who provided much of the foundational research on the human microbiome, focused much of her early work on a type of bacteria, Bacteroides, that could not only become resistant to antibiotics, but pass on that resistance to surrounding bacteria. Salyers was among the first researchers to sound the alarm about the dangers of antibiotic resistance and its potential impact on health. Researchers are actively studying the relationships between various diseases and alterations to the composition of human microbial flora. Some of this work is being carried out by the Human Microbiome Project, funded in the United States by the National Institutes of Health.